In this video, we will be learning about the genetic material and the search for genetic material. So, what is basically a genetic material? Genetic material. Let's try to find out what is basically a genetic material. As we can see, in every family, there are certain resemblances between grandparents to their children and subsequently their children. Now, if we consider grandparents, parents, and children. Now, these children, they will have some similarities to their parents, and these parents will have similarities to their parents, that means to the grandparents. Now, these similarities are due to because of the transfer of genetic material. Transfer of genetic material from grandparents to parents and subsequently from parents to children and subsequently their children, so on. Okay, so this will be carried on from generations to generation. Now, what is the chemical nature of these genetic material? Initially, this particular question, it confused the scientists. Let me change the color. Yeah. So, what was the chemical nature? Scientists basically tried to figure out the chemical nature. They thought it could be either protein, it could be either DNA, or it could be either RNA. But they had no clue which one of these three was actually the genetic material. So in order to find out, there was a scientist, Frederick Griffith, who carried out a series of experiments. Experiment 1, 2, 3, and 4. Now, Griffith basically carried out his experiments using a bacteria and a group of mice. So, this bacteria was basically Streptococcus pneumoniae. Now, we can infer from the name, this bacteria was the causative agent of pneumonia. Okay. Now, Griffith basically took two forms of this particular bacteria. One was the smooth form and one was the rough form. Now, we will see the differences between these rough form and the smooth form and what are these basically. Now, differences between smooth form and the rough form. Okay. So, smooth form was basically, this name was given because of its structure. Its structure was its surface was smooth. It gave a smooth, uniform appearance. But this rough form, it kind of has a non-uniform surface, irregularities over its surface. When these smooth forms were grown on culture, Petri plates, it gave smooth cultures, like smooth colonies. But on the other hand, when these rough forms of bacteria were grown on culture plates or Petri plates, they gave rough colonies. Another difference was, this smooth forms of bacteria, it had a sugar coat over its surface. Sugar coat. This is sugar, or more appropriately, this is referred to as a polysaccharide coat. Now, don't get confused with this polysaccharide word. This basically means sugar. Polysaccharide coat. Now, these R forms of bacteria, it had no sugar coat. Okay. Now, another difference between this S and R was that these S forms are harmful or lethal. Why? Because they caused pneumonia. But these R forms, they were harmless, non-lethal, did not cause pneumonia. Okay, so these are the basic differences between the R forms and the S forms. Contains a sugar coat, does not contain a sugar coat. Gives smooth colonies, give rough colonies. This is harmful, this is harmless, this causes pneumonia, this does not cause pneumonia. These are the basic differences between smooth strain and rough strain. Now, strain is basically synonymous to bacteria only. Strain, whenever I will use the term strain, just know this, the strain is basically the bacteria. Bacteria is being referred to as the strain of bacteria. Smooth strain of bacteria and rough strain of bacteria. Okay. Now, let us look at the experiments which Griffith carried out. Now, in the first experiment, now this is the first experiment. In the first experiment, Griffith took the S strain, S for smooth strain. He injected them into the mice, and then he observed that mice died. Now, quite naturally, we just now saw that S strains were harmful. That's why this caused the mice to die. Okay, and this R strain, these were harmless. That's why the mice lived. This S strain caused pneumonia. That's why mice died. R strain, no pneumonia. That's why the mice lived. Now, secondly, another experiment he carried out was he killed those S strains. He killed. Now, quite naturally, when the bacteria which were causing pneumonia, now they're dead, they are no longer able to cause pneumonia. Not able to cause pneumonia. That's why this time the mice lived. But now, the last experiment, now this was the third experiment and this was the last experiment. In this, what Griffith did, he mixed those heat-killed S strains of bacteria and the R strains of bacteria. This time he saw, now when he injected this mixture into the mice died. Now from this, he actually conferred that, wait, let me just erase this. So, if we just focus on this mixture, we'll see what was the mixture which he made in the fourth experiment. He mixed killed smooth strain plus live rough strain, and this mixture of bacteria, they were able to kill mice. Now from this, he made an inference that maybe some things or some substances were getting transferred from the smooth strain to the rough strain. He just made this inference that maybe there were some substances which were getting transferred from this killed, mind you, this is killed smooth strain of bacteria, and they were getting transferred to the rough strain. Now, he made another inference that maybe these substances which were getting transferred to the rough strain, these substances caused the rough strain to become harmful. Now, initially we knew that rough strains were harmless. Now, he thought maybe after getting these substances, when these substances were added to the rough strain, they became harmful. So, in other words, he inferred that these R strains were getting transformed. These R strains, just erase these. R strains they were getting transformed into lethal forms or lethal S forms. The mixture actually contained killed smooth forms, but now he inferred that maybe there was something, some exchange was occurring between these two strains, from the killed S strain to the live R strains, and that's why these R strains were getting transformed into lethal S forms. Now, why he actually made this inference? Because he was able to retrieve certain live S strains from this dead mice. But initially he gave no live S strains, it was all he killed S strains. But from this dead mice, he was able to isolate live S strains, which also kind of gave him this confirmation that maybe R strains are getting transformed into S. R is getting transformed into S. Now his research or his experiments were carried forward by another group of scientists, Oswald Avey, Colin Mclyn and Mclyn Macarty. They took this heat-killed S strain of bacteria. Now, they wanted to study that what were these substances which were causing the transformation of R into S. They wanted to study, so for example, if I draw over here, if this is the S strain of bacteria and this is the R strain of bacteria, and there is exchange of certain substances from the S strain to the R strain. Now after this exchange, this R strain is becoming, if this is S, this is R, after the transfer or after the transfer of these substances, this R is getting transformed into S. Transformed into S. Now, what are these substances? That's what Avey, Mclyn and Macarty, they wanted to study. What are these substances? So what they did, they took this heat-killed S strains of bacteria. They took these bacteria and they isolated whatever biochemical substances they could find inside these bacteria. They isolated everything, starting from proteins to DNA, RNA, everything. They isolated all the biochemicals from them. And they did another round of experiment. What they did, they treated the mixture individually with protease, RNAs and DNases. And then they observed the process of transformation. Now before jumping into this, let us now see what are these things, protease, DNAs and RNAs. Protease is something which destroys protein. Similarly, RNAs is something which destroys RNA. On the other hand, DNAs is something that destroys DNA. Now they wanted to study the effect of these things on the process of transformation. Now what was this transformation? This transformation was R to S transformation. R to S transformation. They wanted to study the effect of these protease, RNAs, DNAs on this transformation of R to S. They saw that protease and RNAs, they did not affect transformation. On the other hand, when they put DNAs inhibit transformation. Meaning what? When they were destroying the protein and the RNA, transformation was not hampered. R to S transformation was carried on. But when they were destroying DNA, that's when the transformation did not take place. That means something was about this DNA which was causing the transformation to happen. So whenever they were putting DNAs into the medium, DNAs were destroying the DNA, and that's why no transformation was happening. So if you go back to that slide, if this transferring material was, say for example, let me just erase this part. Yeah. So consider if this is protein. Consider if this is DNA. These substances are DNA. Okay. Now, if you are providing protease, protease destroys only protein. It does not hamper DNA. So it will not destroy DNA. Similarly, RNAs will also not destroy DNA. On the other hand, DNAs will destroy DNA. Now, if DNA gets destroyed, no transformation can take place. No transfer will take place, and that's why no transformation will take place. Okay? So, that's why Avey, Mclyn and Macarty, they came to this conclusion that DNA was the transforming material, or DNA was the genetic material.